All right. So, hi, everybody. Welcome to our introduction to computational approaches for cell biologists. So, my name is Leonard Harris. I am an assistant professor of biomedical engineering at the University of Arkansas. And with me is Alex Lubbock. He's a research assistant professor in the Department of Biochemistry at Vanderbilt University School of Medicine. We work together for a number of years. I was also a postdoc at Vanderbilt before I started here last year at Arkansas. And so we're going to give you your intro to computational approaches. So I know that, the idea of this session is supposed to be, to talk to cell biologists. So people might, interpretation of a cell biologist is somebody who has little to no math or computational background. So that's the level that I put together these slides. So I have quite a bit of experience actually working with experimentalists, and I'm going to show some of the slides that I've given in other contexts to experimentalists, talking about what the basics of modeling, that sort of thing. And then I'll go through a little example from my own work before Alex will then take over and do a hands-on tutorial on how to think about a model, build a model, use some software, that sort of thing. Okay, so let's get started. Move forward. Okay, so let's start way at the beginning. Okay, so the first question that comes up always is, why exactly are we building a model? It's a more common question than you might think. Or maybe you have had this question yourself. That might be why you're actually here at this conference, right? So here's a quote that I really like from very famous genius, John von Neumann. He says that the sciences do not try to explain. They hardly even try to interpret. They mainly make models. By model is meant a mathematical construct which, with the addition of certain verbal interpretations, describes observed phenomena. The justification of such a mathematical construct is solely and precisely that it's expected to work. So this is a really interesting take, right? He's basically saying that science is modeling. And so the way, and he's talking, of course, he's a mathematician and a physicist, so he's talking specifically about mathematics, which I agree with, but I think another way or an important way to interpret this quote is that since all science is modeling, all of you, even if you've never built a model before, are also modelers. You just maybe model what you imagine is going on inside of these cells, or what you think is going on inside of cells or tissues or whatever in different biological systems inside your head. When you draw a diagram in a textbook, or you see a diagram in a textbook, or you draw something on a board, or in a paper, that's a model. The only difference is it's not formalized in mathematical language. And so von Neumann says you must turn that into mathematics in order for it to be, what he considers science. And that's what we are here to do today, right? That's the point of this conference, is to try to take all of this biological knowledge that's out there and turn it into some kind of formal mathematics. Okay. Another quote that you may have heard before, if you haven't, if you keep working in this area, you will hear over and over and over again, is the quote from a guy named George Box, where he said, all models are wrong, some models are useful. Okay. I want to caution that many people focus on the first part about all models being wrong without recognizing or emphasizing the second part, that some models are useful. So when they say all models are wrong, all that means is that a model, a mathematical model, is a representation of a real system. It is not the real system. So because it is obviously not the actual system that you're studying, there will be things that will be left out. That is inevitable. Nevertheless, there is a tremendous amount that you can learn by building a model, a replica of a real system, and those models that are able to explain and make useful predictions are the ones that are very useful. Okay. So here's an example, a very simple example that I often like to give to like first year systems biology students, for example. So forget about biology for just a second, and remember back to your high school, physics classes or science classes where you learned about Copernicus, right? Copernicus was, lived in the 1400s, 1500s, and he had this crazy idea that maybe the Earth was not at the center of the universe. Maybe, in fact, it was the sun that was at the center of the universe. To them, the solar system was the universe. And the planets moved around the sun in concentric circles, right? This was a model of the universe. Now, a couple 100 years later or so, Galileo, who I'm sure you've heard of, had revisited this idea, right? But unfortunately for Galileo, this was a very taboo topic at the time. So he was thrown in prison for this crazy idea. However, his contemporary Kepler, who was far away over in Germany, was able to pursue this idea and free of prison, free of the law, of legal threat. And so what he did is he said, okay, you know what? Copernicus was wrong. The sun is not at the center of the universe and the planets move around it in concentric circles. Actually, the sun is more like the foci of a ellipse. And so the planets move around the sun as ellipses, not as circles, okay? So here's one very famous example of a model in the case of Copernicus that was wrong and yet immensely useful, okay? Now, also, this model by Kepler is an example of a phenomenological model. It explains something. It is predictive of the future, right? You can use this model to predict. If you know where the planets are today, you can predict where they will be in the future. You don't know why the planets are moving in concentric or in ellipses around the sun, but nevertheless, it's very useful in making predictions and understanding the nature of the universe. Now, 100 years or so later comes Newton. And so Newton changes the entire game. Newton defines or invents the theory of gravity, right? And says that two masses, they interact with each other with a gravitational force that is proportional to the product of the two masses and inversely proportional to their distance. And so Newton's theory of gravity now is an example of a mechanistic model. It explains why bodies move in a gravitational field the way that they do, right? However, a couple 100 years later, Einstein comes along and says, well, Newton was wrong. But of course, Newton's model was also very useful, right? So here again, just using some well-known historical examples are examples of models that are extraordinarily useful. Nevertheless, they were wrong. And we also have examples here of phenomenological models and mechanistic models. So in the context of biology, these phenomenological models are analogous to like bioinformatics. And this often comes up when we talk about systems biology. What is the difference between systems biology and bioinformatics? So I'd say bioinformatics is kind of part of the data side of things. And it's part of modeling, but it's on the phenomenological side. It's looking at correlations between data without really trying to have a deep understanding of why one protein correlates with one clinical outcome, for example, right? Now, Newton and Einstein are more analogous to like a theoretical biology approach. So trying to build mechanistic models that explain why certain proteins within interaction networks drive certain phenotypes, for example. And to me, the combination of these two, bioinformatics and theoretical biology, is what defines this whole field that we work in of systems biology. Okay. So that is systems biology in a nutshell, without even talking about biology yet. Okay. So the second thing that comes up often when I work with experimentalists is this question: how much data do you need to build a model? That always happens. And my answer to this question is always whatever you got. Okay. Now that might sound strange. So let me explain using a very oversimplified example. Okay. This is what I mean. So let's imagine an extreme case where we literally only have one data point. Let's say we've got one protein concentration at one point in time, and we're asked to build a model. Well, we could certainly build a model of this if we know, let's say that we know concentration is 0 at time 0. We could build a model of that data point. It could be a linear model. But we could also build another model, right, that looked like this, or a third model that had some kind of behavior like that. And so the point is that building models is not hard. In fact, building models is extremely easy. You give me one data point, or you give me any data you have, I can build a model. The problem is having any confidence that the model that you've built relates in any way to reality, right? Because I can have all sorts of different possible explanations for this one data point that you've provided, or this set of sparse data, say that has been provided. So in one of the talks earlier, and actually in the conversation afterwards, there was some discussion about what is broadly called model selection. So the idea of having multiple different models and testing how each of these different models compares against a set of data, and using some sort of Bayesian or other type of criteria for ranking the models and giving you an idea of which one is most likely to be the best one, or the one that to correspond to reality the most. Okay, and so that's a very important part of this whole process. So model selection techniques, for example, these acronyms, this stands for the Akaki Information Criterion. This BIC stands for Bayesian Information Criterion. These are just a couple of examples of metrics that people use given a set of data. You're given your available data and a collection of different candidate models. You can rank them based on these different types of metrics. And as I said, there are many different types of these criteria, and which one you use is kind of depends on who you ask. You've got to be kind of an expert in this field to know. And we'll get a little more, I'll talk a little bit more about that in just a minute. Okay, but now let's move on. So let's say, okay, now we've gone and collected a second data point. So now we've got a data point up here, we can ask the question, which of these models can explain that data point? Well, this linear model would not be able to explain that data point. This more quadratic model would be consistent with that data point, and this oscillatory model would not. So once we do that, once we collect more data, we can immediately throw out models 1 and 3 as candidates. They're no longer almost, there's almost no chance that they are correct representations of reality, because they can't explain the new data that we've collected. So model building is an iterative process. And I think for me, this is like the most important thing for students and for experimental collaborators to understand, is that we don't just take data, build a model, write a paper, and then that's it. I mean, practically we do that. But the reality of the process is that this is an iterative process. So we take the data that we have, we build the best model that we can. We maybe put together a collection of different models and come up with the best hypotheses and say, okay, given what we have, it might be models A, B, or C. And that's as good as we can do. But now we can use those models to make predictions, which we can then use to trim down the possible candidate models. We may also generate new candidate models in that process, and we repeat the process over and over and over again until we feel comfortable that we have a model that can answer whatever questions we are asking. And in some sense, this is a never-ending process, because like I said, all models are wrong. So you can always get better and better and better. It is just up to you to decide when the model has become useful enough to answer whatever you are whatever questions you're trying to answer. Okay, so with that background, this is something that I like to call the 6 easy steps to building a computational model, and easy in quotes. Okay, so let's start with step number 1. This is something that everybody can do. This is something that, in fact, the biologists and the experimentalists need to do when I work with them, is to list out everything that you know. So you need a comprehensive list somehow of all experimental data, everything in the literature, whatever you think whatever you understand or think you understand about the system. We need a comprehensive list of all of that. Okay, now with all of that information, we need to draw some type of a diagram that is able to kind of summarize all of that information in some pictorial form, right? So these first 2 steps anybody can do, and like I said, is actually, there's a more, it's the sort of thing that the biologists and the experimentalists need to do for the modeler. Now, the 3rd step is to now take this diagram and formalize it into some kind of mathematical form, right? Into some kind of model. And that's where the modeling expertise comes into play. So, and I'm going to talk a little bit more about this without going into a lot of detail, but sometimes you can do this yourself, you know, if you know a little bit of math, or you know how to use a certain tool, maybe through a workshop like this or something. Sometimes, though, it might require a real expert who works in this area, so through some type of a collaboration. Now, once you have the model in a mathematical form, the next step is to investigate the behavior of that model in silico. So what I mean by this is the model itself is now the object of your investigation. The parameter values that you choose are going to determine the behaviors that this model produces. So certainly what I like to do is to look at how the behavior of this model changes as I vary different parameter values in different ways. I just want to see what this model is capable of doing. I like exercise the model, push it, pull it, squeeze it in all sorts of different directions to see what the model is capable of doing. Because like I said, this model is, we hope, is some sort of representation of reality, but it is the model itself that is actually the object of our investigation. And then, so we have to study that, and then we have to make the argument that what that model is doing has some connection to reality. So then the 5th step is to actually compare the model to experimental data. And so this is where we have to do things like estimate the parameters by comparing model outputs to experimental data, and then also using the models that have been fit to the experimental data to make model predictions that can then be tested by follow-up experiments. And this can be used to drive experiments. And so I always like to emphasize this point that I think a lot of people often jump to step 5 without going through step 4. And I think that's a mistake. I think oftentimes you run into issues where all of a sudden your model is behaving in ways that you didn't expect. After the fact, after you fit it and start making predictions, all of a sudden the predictions seem strange because you don't understand exactly what the model is capable of because you haven't gone through this 4th step. Okay, and then you just repeat. So as I said, you use your model or models to make predictions that can then drive new experiments. And now that you've got new experimental data, or there might be new data from the literature, you now know some new things. So you have to add that to the list or modify your list somehow. You may have to modify your diagram, and then you may need to reformalize it, change your model structure, and then redo this whole process. And it's an iterative process. It just keeps going over and over again. Okay. If there are questions, I guess, I don't know. I guess maybe I'll. Yep, go ahead.
Yes, good evening. I think I would like to clarify the fourth step about investigating the behavior. Do you mean that we are supposed to check, for example, extremomes and like limiting factors of the function itself? This is the main idea of step 4.
What was the first thing you said? Sorry.
So just study function as a mathematical function. So what are the extremomes of the function, the limiting factors that we introduce in the beginning of the model, right? Is this the idea?
Yeah, I would say so. At least within some sort of physiological ranges, you know, of the parameters.
Oh yes, of course.
Yeah. But yeah, exactly. Because like I said, often I think there's a tendency to put a model together and then to immediately fit it to data without really having any intuitive understanding of what the model is capable of doing. And I think you run into problems when that happens. So it's good to have an idea of, you know, if this parameter is very large, you tend to see this type of behavior. If it's small, you see that kind of behavior, that sort of thing. Like make a phase diagram, that kind of thing. Yep.
How do you know when it's not right? I still am hung up on number 4.
So you won't know if it's not right until you make predictions in step 5 that you can test.
Okay, thank you.
Yeah. So step 4 is just about getting a feel, basically, for what you're working with. Yeah. Okay. So I'll keep going. So this part though, step 3, right, formalizing the diagram mathematically is really kind of why we're all here. This is the idea of connecting modelers and biologists, right? So I'm not going to go into a lot of detail about this. I'm just going to put up 2 very quick slides. One is that whenever you talk about this sort of thing, about how to choose a modeling method, you will often see diagrams like this on the left, where people will plot some kind of a time scale versus a length scale, and you'll see this sort of thing, right? So you have genes, proteins, organelles, cells, tissues, organs, all the way up to the length of the body. And, you know, there are different modeling methods that are applicable at these different scales. And you'll have to kind of understand which ones apply where. And that's sort of what is shown here in this pyramid cone thing here in the bottom right. But one thing I wanted to point out here, so is that, you know, so they start all the way down here at the quantum level, if you're really looking at like single molecules inside the DNA, up to the atomic level. So down at the quantum level, you're talking about, you know, Schrodinger's equation. You get up into atoms, molecules. This is it's a little hard to see, but this is molecular mechanics, molecular dynamics, also partial differential equations, stochastic methods like Langevin equations, Markov dynamics, and Gillespie simulations, all down here. Then when you get up to the level of proteins here, this is ordinary differential equations, and this is reaction diffusion equations, which is also partial differential equations, also apply here. At the cellular level, again, ordinary differential equations, partial differential equations, or you can have agent-based models, discrete agent-based models. At the level of tissue, again, partial differential equations, cellular automata, CA. So you'll notice in particular, things like ordinary differential equations and partial differential equations can be applied at many different scales. So it's not as simple as saying, okay, at one scale, there's this method, and another scale, it's this method, another scale, it's that method. A lot of the methods can actually be applied across the different scales. So it's not a simple answer. Again, why you have to have some expert knowledge, and an expert collaborator will help a lot in this regard. And so here's just a very non-comprehensive list, okay, of some different modeling methods here on the left, and then I've listed out some software here on the right. So I'll just go through them really quickly. So for mechanistic modeling, which is really what this is my background, so that's why the list is longer. But so things like molecular dynamics, Brownian dynamics, that's for things like protein structure, protein folding simulations, that's what people use there. Stochastic simulations can be applied at many of these different scales, cell level scale or intracellular simulations. So things, stochastic simulation algorithm, there are things called tau leaping methods, which are accelerated methods or master equation approaches, etc. I mentioned ordinary and partial differential equations that can be applied at many different scales. Agent-based models are popular, especially for like cell or tissue level simulations where you have cells that are moving around and sensing their environment, and you can define some sort of simple rules that define their movements and behaviors. And then things like logical models, which include Boolean models that kind of are inspired by circuit models of network dynamics that can be applied to cells, and also things like in particular gene regulatory networks. And then there's also data-driven models. So I mentioned the phenomenological models, the bioinformatics side of things. These are very important. Again, not my area of expertise, but is something that I'm getting more involved in. So I wanted to just mention them. Things like multivariate regression and certainly machine learning, artificial intelligence. As I mentioned earlier, you know, these give you correlations. So these are models and they are predictive. They're not, they don't answer the why. So connecting data-driven modeling with mechanistic modeling is a very interesting area of research that I'm personally interested in. And then in terms of software, molecular dynamics, a very popular tool that's used for biological applications is CHARM. There are some stochastic simulation software toolkits out there, StochKit and StochSS, the Stoch simulation service. Also some well-known simulation tools, CoPosse, the virtual cell, and Tellurium. There's a type of model of kinetic modeling that Alex and I are very familiar with, and Alex's tutorial is going to use this software called PySB. Also a related tool called BioNetGen that is part of PySB and I have worked on in the past. There are agent-based tools. PhysiCell is a pretty popular one. NetLogo is an older one, but is also still quite popular. For Boolean logical models, BooleanNet, GenSim are a few. And then for data-driven models, there's SPSS, which I think is an IBM tool, also TensorFlow. And then there's all sorts of packages out there for both mechanistic as well as data-driven modeling in R and Python. MATLAB too, probably. So the main point here, though, is that some of these methods and tools are easier to use than others. Certainly, some of these tools are designed to target people without much of a biological background. Other tools are more for experts. So it depends. It depends on what your application is and your personal background as to whether you'll be able to use these yourself or whether you'll need an expert collaborator. But there's lots of options out there. Okay, so what I'm going to do then is I'm going to go through just maybe in about 15 minutes, an example out of my own work that goes through each of these steps. And I'm not going to spend too much time on the biology part of it, although I will present it just so that you see that there is real biology behind this. But I want to give a practical example of how one actually does this type of model building exercise. And then when I finish, obviously I'll be happy to answer questions, and then Alex will take over and do a practical tutorial. So, and by the way, if you know, of course, if there are any questions as I'm going through it, please feel free to interrupt me. Okay, so this is a project that I have been working on now for a little while with some collaborators that I have at the Vanderbilt Center for Bone Biology. It's studying tumor-induced bone disease. So very simply, when tumor cells metastasize to the bone environment changes their signaling. And this typically results in net bone loss, which is broadly called tumor-induced bone disease. It leads to very brittle bones, cancer patients, they end up with a lot of bone pain, and they can get breaks more easily. It's not a pleasant disease for sure, pleasant condition. So there are drugs, bone-targeted agents that can reduce bone destruction, and they can improve quality of life. However, there are no treatments that can both reduce bone destruction and cure reduced tumor burden. So even though the quality of life is improved, there is no improvement in the actual survival of patients. So there is a real need for new treatment strategies. At a molecular level, tumor-induced bone disease is due to the secretion of tumor-derived factors. For example, this hormone, this is parathyroid hormone-related protein, PTHrP, which secretes out of tumor cells, and it interacts with osteoblasts to drive expression of rank ligand on the surface of osteoblasts, which then activate osteoclasts, which are the cells that consume bone, consume the bone matrix. So basically, the tumor cells secrete factors that knock the natural bone production and consumption processes out of whack. And so the osteoclasts consume more bone than they normally would. And in the process of consuming the bone matrix, they release TGF-beta, and TGF-beta then feeds back onto the tumor cells and drives further expression of PTHrP. And my collaborators have focused on this gene GLI2, which they suspect is a main driver of this whole process. So this is what they refer to as a vicious cycle, where the tumor cells release this PTHrP, which then ends up driving more expression of itself. And so the system just continues to consume bone until either you put in something to prevent it or the bone is completely consumed.
If you're the tumor, it's just a positive feedback loop.
Yeah, exactly. That's right.
But let's not make value judgments.
That's right. Okay, so here is just one very quick slide about some very basic experimental results that are at the heart of this model. So here on the left are these three proteins. So there's PTHrP, which you saw on the previous slide. There's this GLI2 protein, which I said is they believe is central to this whole process. And then there's also integrin beta-3. Integrins reside on the surface of the cells, right? And so you can see right here, this is all mRNA. And along the x-axis, these are different substrate stiffnesses. And so at low substrates, so on soft surfaces, there is a very low expression of each of these. And these three lines are for three different bone metastatic cell lines. And so on soft surfaces, there's low expression. And you can see that as we increase the stiffness, the expression goes up. So there's clearly some type of a correlation, right? Some kind of connection here between PTHrP, GLI2, and the integrin. And now they've also done these FRET experiments, which can basically just see whether or not different molecules on the surface are co-localized, whether they're close or binding with each other. And so what they saw is they looked at TGF-beta receptor type 2 and its interaction with integrin beta-3, the same protein down here. And what they saw is that on soft surfaces, this is a fluorescence, which is just a measure of basically the signal coming off of this. High fluorescence means that there is a strong co-localization. On soft surfaces, there's basically no interaction. On hard surfaces, there's a very strong interaction. So these are being co-localized on hard surfaces while they are not being co-localized on soft surfaces. And so we know this co-localization and also is also then correlated with high expression of PTHrP, GLI2, and the integrin itself. Okay, now, so to go back then to the six active, or to the six easy steps, step number one is to list out everything that we know or that we think we know. So I have a number of bullet points here. I'm not going to hang on them too much, but I'm just going to pull them up one at a time. And these are bullets that my collaborators gave me when I started this whole project with them. So the first thing is that PTHRP is known to be expressed in patients with bone metastases. For example, 92% of patient samples in bone metastases, as opposed to 17% in patients that have non-bone metastases. They also know that GLI2 is expressed in bone with increased rigidity, as you just saw. GLI2 also is known to regulate PTHRP in a hedgehog-independent manner. So GLI2 is part of the hedgehog signaling pathway. However, this is acting in a non-hedgehog context. GLI2 knockout mice are known to have reduced tumor burden and bone destruction. It's also known that TGF-beta regulates GLI2, but it is not bone specific. And then, as you just saw on the previous slide, as rigidity increases, TGF-beta receptor type 2 and integrin beta-3 co-localize. They do not co-localize on soft surfaces. And this stimulates mechanical signaling, specifically through proteins FAC, ROC, and others that they have studied over the years that results in increased GLI2. And then they've done a number of different intervention studies. So for example, they have a TGF inhibitor called 1D11 that is known to reduce GLI2 expression in vivo. However, interestingly, it does not reduce tumor burden in vitro, does not reduce tumor burden in vivo once the tumors are established, although it can if it's given prior to tumor establishment. So we still do not understand why this is the case. GLI2, they also have another drug, GANT58, which is a GLI2 inhibitor, which reduces bone destruction, but does not eliminate tumors in vivo. And they can inhibit integrin beta-3, which blocks mechanical responses of tumors and prevents bone destruction, but not tumor growth. Okay, so this is a somewhat comprehensive, not an entirely comprehensive list of everything that we know about this system. So step 2 then is to draw a diagram. And so this is something then that they helped me with. I sat down with them at a board, at a whiteboard, and we kind of worked this out, and they helped me understand exactly how to lay out this diagram. So very briefly, up here, this is the cell membrane. Down here, this is the nuclear membrane. So this is the nucleus down here. And the idea is that we have TGF-beta receptor type 2 and the integrin, alpha V beta-3 integrin that can interact reversibly. TGF-beta can reversibly bind to the receptor. Then it's known that TGF-beta receptor interacts with SMAD, phosphorylates SMAD, and SMAD can translocate into the nucleus and act as a co-transcription factor for GLI2. Then off of the integrin, it's known that there is a cascade of events that goes through row A, ROC, MEK, and P38 MAPK, and phosphorylated P38 MAPK acts as a co-transcriptional activator with SMAD to activate GLI2. GLI2 then acts as a transcription factor for the integrin and for PTHRP. So PTHRP gets driven, this is expression PTHRP, which would then presumably get secrete out of the cell. The integrin comes all the way back up here where it interacts with the receptor. So when you look at this at a broad sense, what's happening is there are two signals that are converging on GLI2, and then there's a feedback that's coming back up here. And so anybody that knows or that works in this area, Professor Stone, for example, would know that once you see a feedback loop like this, your model is going to have some interesting behaviors associated with it because of these things like bistability, that kind of stuff are features of feedback loops. Okay, and then as I mentioned, they've done a number of experiments using TGF inhibitor, GLI2 inhibitors, as well as applying growing these cells on soft and hard surfaces. And so the idea is to reproduce or do in silico experiments of those types with this model, and then see how the results compare to their data. Okay, so in terms now for step 3 is where you have to formalize it in terms of math. And so, like I said, this is where, as the experimentalist or the biologist, you may need some help from an expert to do it. So I'm going to go through just a couple of very quick slides without going into a lot of detail again to show how I put this model together. There's not a lot of math in here, but just very basically. So there's a couple of basic interactions here. So one is reversible binding. For example, the like ligand reversibly binding to the receptor. We also have the integrin reversibly binding to the receptor on the surface. SMAD, phosphorylated SMAD can reversibly bind to the receptor. And then they're also modeling the interaction of the drugs and as a reversible binding event. So we can just model it as a simple reversible reaction set. A plus B goes to an AB complex. We modeled the rates of these reactions just using the simplest form, which is known as mass action kinetics. And then we can define out of this what's known as a dissociation constant, which is just the ratio of the reverse to the forward rate constant. And we can keep track of this dissociation constant, and we can see how the behavior of the model changes as we vary this value. For this catalytic activation and phosphorylation cascade, we model it in a very simple way. We just say that we've got one of these proteins, for example, row A, that interacts with some, say, catalyst X. For row A, it would be the integrin. And just by interacting with it, it activates it, turns it into some other form, A star, or maybe in this case, MECP. And again, and then it can also deactivate in a simple way. And so again, we use simple mass action kinetics to do that. And as I mentioned earlier, when the integrin is bound to the receptor, there is a stronger signal that must be coming through this because there is a higher expression of all those other proteins. So in this version of the model, the way we're going to handle that is by saying that the rate of activation of row A is increased when the integrin is bound to the receptor. Okay. And then for the last one, for gene expression, this one is a little more complicated. I mentioned GLI2 drives expression of the integrin gene. It can also drive expression of PTHRP. So a kind of common way to model this is as a multi-step process. We have this transcription factor T. We assume it can form an oligomer of some size N. It may be a dimer, maybe a trimer, you don't know. But we imagine that it forms in one step, it forms an oligomer, and that oligomer can then bind to the promoter site of the gene to form a complex, and then the bound form of that gene can then produce mRNA. And the thing is that we can then apply some assumptions to this to simplify this down, and we can end up with an effective rate for production of the gene, of the mRNA, in terms of something known as Hill kinetics, or this is a Hill kinetic form. So when people write these models, you will often see these types of terms thrown in there. This is the basic mechanism from which they come. Sometimes that's explained, sometimes that's not. But that's where they come from. This is what a Hill curve looks like. It's kind of like a step function. As you make this exponent larger, it gets steeper and steeper and steeper, so it becomes more switch-like. And then I also mentioned that for GLI2, there's actually two proteins that co-bind to drive it. So there's a way to just, there's an equation for just generalizing this. So this is a general Hill form. Okay, so that's all the math that goes behind this model. Every one of these reactions is modeled in one of these three different ways. And so now with that, the fourth step is to exercise the model and see if it has some interesting behaviors. And so I'm going to show just one very short animation to give one very interesting example. And I have a little animation over here on the right to help kind of guide the idea. So as I mentioned, right, so if you have the thickness of these arrows is supposed to represent how strong the binding is. So in this case, there's a very weak binding between the integrin and the receptor. So if there is a very weak binding here, then row A is going to get activated primarily off of the integrin that is not bound to the receptor. And that's going to send a signal together with SMAD that's coming off of the receptor down to GLI2 and try to drive expression of GLI2, which would then feed back. But when we actually run the simulation, nothing happens. GLI2 does not turn on, it just stays off. Okay, so now if we bring in a mechanical force, which is modeled by increasing this rate constant, increasing this interaction, then what that means is that row A is now going to get activated at a stronger rate at a higher rate off of this bound integrin. And that leads to a stronger signal, a stronger feedback, which causes GLI2 to turn on. Okay, so that was good. That was not unexpected. So I showed this result to my collaborator and he said, okay, well, that's interesting. What would happen if you took the force off? So if we remove this force, everything would go back to here. So your first instinct would be everything would just shut off, right? But what actually happens is that while the level comes down, it doesn't go all the way to zero. It actually stays elevated. And at first, I didn't understand this. This is a non-trivial result. I mean, at least for me, it was at first. And it took me a little while to intuitively understand why this is happening. And so the answer, at least the analogy that I use is it's like a spark plug in your car. You need a spark plug to get your engine started, but once it's started, it's self-sustaining. So even though this weak signal that comes off of the integrin that is not bound to the receptor is not strong enough to turn the GLI2 level on, it is enough to keep it on at an elevated level once it's been turned on. And so this was actually really interesting because this feeds into this idea that this is a type of vicious cycle, that once this process of tumor-induced bone disease gets activated, it's very hard to reverse it. So that's one important and interesting observation we got. And this is not based on any type of parameterization against any type of data or anything. This is just a behavior that the model exhibits naturally.
Leonard, can I ask a question on that
Sure.
slide?
Yep.
So I may have blinked. Did you just draw in the red arrow between GLI2 and the integrin? Or was that there before?
This one?
Yeah.
That was there before.
Okay, so it seems to me it should be a somewhat trivial result because GLI2 is, I presume, activating the ability of the integrin to activate Rho. So there's
Yes,
the feedback. That's why it's persisting, right?
that's right. That's right. But it's the previous case where it was it was just low, it was off. It's exactly the same condition. It's just that the initial conditions were different.
Yeah.
Yeah. So that's, we can see that here. So then I just did a bifurcation analysis, which just shows this. So this, I mentioned KD earlier as the, yep.
Excuse me. Can we go to the previous slide? I just
Sure.
want to ask about, you say that in the previous slide, when it was just a zero line, the conditions are the same, but the GLI concentration should be, yes, but the GLI concentration in the cytoplasm, it's zero, right?
Mm-hmm.
And after we have the initiation, the result that it doesn't go back just because the GLI is still in the so it already activated the integrin, right? And
Yes.
the process is not, it's no more the same. Right.
So the starting conditions are different. So now it's got the same rate constants, the same activation rate here, the same rate of expression of GLI here. is all the same. It's just that in the first case, yeah, we start with no GLI and low levels of integrin. And in the second case, because we've turned GLI on and we have high levels of integrin, then that's the difference, is that there's different initial conditions. And so the signal that's going through here is enough to keep it elevated because we have started at a higher initial condition for these initial levels for these proteins.
Okay, thank you very much.
Yep. Yeah, so like I said, this is it depends. So, you know, if you understand dynamical systems, it is a bit of a trivial result, maybe, because you understand that feedback loops support these types of dynamics. And so after thinking about it, yeah, it made sense that it wouldn't go all the way down to zero, but it's not necessarily something that I anticipated. This was something that came out of running the simulations, and then I had to think about it after the fact to realize that it made sense. So a lot of times
I think the thing is that if we replay this tape, which we will, you said initially it's the same initial condition. That's what we were reacting to.
Oh, no, I'm sorry. It's the same like rate constants.
Yeah.
Right, right.
In fact, the level of GLI2 is the key factor.
Yes, yes, absolutely. Yeah, I didn't mean to imply that it was the same initial conditions. It's just the same network with the same rate constants, but with different initial conditions. And so it achieves a different steady state. So the system is basically bistable in that way. And then that's what I've quantified here in what's called for everyone else, what's called a bifurcation analysis. So up here at these large values of KD, this is very low interaction, very weak interaction between the integrin and the receptor. So you can, and this is the GLI2 level over here. So with a very weak interaction, there's zero GLI2 until we hit this threshold. And then boom, around 2000, and then boom, it turns on. And then we reach a level of around between 11 and 1200. But now if we start here and we go back in the other direction and we begin to remove the force, then it doesn't come back this direction, it goes this way, it follows this branch. So this is broadly, this is termed hysteresis, where it doesn't follow the same direction. So it keeps coming this way, and you can see it's interesting that it doesn't come back down to zero ever. It ends up staying elevated up here around 800, which is what was on the previous slide. Okay. Okay. And so now that's really where the state of this is, and so this is also why I kind of emphasized that step 4 earlier, right? Because I think that there's a lot that you can learn by just taking a model and playing around with it, moving the parameters and seeing what the model is capable of doing. You can gain a lot of insights just by doing that without even yet looking at data. So now what I'm working on with a student in my lab is to now actually use this model and compare it to experimental data from my collaborators at Vanderbilt. So they have Western blots and qPCR, which is a little hard to work with. So we're trying to figure that out. But the idea is to now use the model to fit it to this data. And so that now, instead of having these kind of qualitative predictions, we'd like to make actual quantitative predictions. And I have a collaborator here at the University of Arkansas who works with liquid crystals, and he can do things like he can tune stresses on the surfaces of cells by changing the structures of these liquid crystals, and we can see if in fact the expression levels of GLI2 and other proteins change in the way that the model predicts. So that's where we're moving. And so, like I said, we want to test predictions, for example, this partial GLI2 reduction after force removal. And there are a number of other predictions that we've made that are also non-trivial in response to TGF-beta inhibition and GLI2 inhibition. And then step 6 is to rinse and repeat. So basically, after we collect this new data and see whether or not our predictions are able to are borne out in these experiments, then we will likely need to update our model to try to understand why we're missing whatever we're missing. So perhaps we'll need to add in some more molecular species. For example, maybe we have to add in all the different variants of SMAD, since right now the model just has SMAD, but there's SMAD2, there's SMAD3, there's SMAD4. There's also a host of other pathways that are involved in tumor-induced bone disease. The Wnt pathway, the hedgehog pathway, the EGFR pathway, and there has to be a lot of interactions within the microenvironment as well. So we're all looking at creating a companion model that includes tumor cells, osteoblasts, osteoclasts, immune cells, that sort of thing, that can also be hopefully connected with this signaling pathway model. So there's lots and lots of stuff that we can do on top of this. Okay, and so that's where I'll finish off. I'm happy to answer any more questions, and I'm also ready to hand it over to Alex for the practical hands-on part of the tutorial.
Yeah, I have a question. Could you explain in a little more detail what you referred to here, but you were referencing what came up in one of the other presentations, the project presentation, which is the probabilistic analysis, wherein you have multiple different models and you're trying to determine which is the best fit at this point. And how does that work? I don't have a good idea at all.
Yeah, I mean, like I said, there are many different ways that people try to do that, but I mean, I can tell you just at a very high level, you know, the idea is that it's a kind of an Occam's razor. That I think is the best way to think of it. It's an Occam's razor argument. So what you want is you want the simplest model that explains the data the best. So, you know, you could have a very complex model with lots of different parameters that fits the data perfectly, or you could have a much simpler model that fits the data a little bit less than perfect. And if you were just to say use an R² value or something, then you would choose the more complex model because it fits the data better. But practically speaking, we, intuitively we understand that actually the simpler model that has maybe a slightly worse fit, but is far simpler and has a lot fewer adjustable parameters, is really the better one. So it's the argument that you want the simplest model that explains your data the best, but not too simple. That's the idea.
That makes sense, but I think I'm hung up on, well, I could eyeball that, but you're talking about something much more formal.
Yeah. I mean, so there are lots of, there are different formal ways of doing this. So for example, there's something called an Akeki information criterion. It's, I don't know, it's developed 20, 30 years ago by Akeki. He has an equation based on some sort of statistical argument, and it takes into account the number of parameters in your model. So it takes the fit, like an R² fit, and then it subtracts, it has a penalty term that depends on the number of parameters in your model. So that's one way to do it. There's also Bayesian information criteria, which is something that's sort of related, but not exactly the same. Alex is also knows a lot about these things. You can chime in if you want, Alex, but I mean, there's more sophisticated Bayesian techniques for doing this sort of thing.
Okay, that's good, thanks.
Yeah, that's basically right. I think, Leonard outlined the main points there. It's something that balances the likelihood function, which is usually defined as the goodness of fit versus some experimental data, and then balanced with the number of parameters in the model. So you balance those two factors according to a specific formula. So are there any more questions for Leonard before we switch over to doing my slide deck?
I think you're up.
All right, great. All right, so hopefully you can see those slides.
Looks great.
All right, so just a few more slides. I've only got a handful, but essentially we're going to set up an activity where we're going to do a breakout room and basically follow the first few steps of the modeling exercise that Leonard outlined. So he outlined these 6 simple steps. We're going to do the first 4 of those today and just apply those to a simple system. So I will kind of go through a couple of rudimentary examples just to, I know Leonard's kind of shown you a worked example, but I'll go through some sort of almost trivial examples and then we'll apply it to a different system, as you'll see. So like if you wanted to have a really trivial example of a dynamic model, right, you could just say we're going to synthesize X, some protein. So when the first step of Leonard's pipeline, we write out everything you know. So we just got one. We often call these molecular species in a model. You can also see them referred to as classes. So we just have this one thing called X. And facts about the system. X is being synthesized. For the purposes of the model, we're just saying it's being made out of thin air at some rate K. So it's not a very interesting model, but it should, it's very simple to get to grips with. So you'll see in a lot of models, they use this kind of empty set symbol to define out of nothing or to nothing. So this is de novo synthesis of X at some rate K. So this is a very simple diagram of what's going on. And then we can characterize this with one differential equation where the rate of change of X over time is just given by this value K. And then if you run this through a simulation tool or an ODE integrator, then you just see the quantity of X is increasing over time. So pretty straightforward, not very interesting. Let's move on to something a little bit more, with a little bit more detail. So many of you might have seen this Michaelis-Menten model. So the idea of this model, again, we'll kind of write down the facts about the system and do a diagram for it. That you'd have some substrate, some protein that is catalyzed by some enzyme. They reversibly form some complex together where the enzyme and the substrate are bound, and then the enzyme dissociates, leaving the substrate transformed into something else, which we'll call the product. So facts about the system: this free enzyme reversibly binds to form the complex with some forward rate and some reverse rate, and then this complex dissociates to a free enzyme and product with some rate, which we'll call kcat. So if we're drawing a diagram of this, it looks something like this. Enzyme plus substrate forms this complex, forward rate, reverse rate, and some catalysis constant where the enzyme dissociates. And of course, now that this enzyme is free again, it can go back and bind with more substrate. So you can write this down in a set of 4 differential equations, which are shown here. So you don't need to worry too much about the kind of the detail of those equations, but they can be derived from this reaction-based model. And this is one of the key sort of points to grasp, is that writing something as a set of reactions and a set of ODEs is sort of an interchangeable way of describing a system. And usually it's easier to kind of think about it in terms of these reactions. So this is the ODE formalism. And again, you can kind of put this into a simulation platform or an ODE integrator and look at the concentration changes over time. So the total amount of enzyme in the system doesn't change. That's the blue line. The free enzyme decreases as it binds to form this complex shown in purple. And the amount of substrate decreases over time as initially a lot of this complex is formed. And then eventually you start to get this product starts to be formed over time. So the dynamics of this are going to depend on the exact values of these constants and the initial concentrations, but that gives you a rough idea about the dynamics of this system.
So I have a question.
Yes.
When this output, you're referring to it as like a generic integrator or whatever, but I mean, how does that work without, I mean, you're not putting in the rates. So how are we getting these generic curves?
So this particular graph uses some assumed sets of rates. So these are for specific values of these. So one of the things that we're going to do today with the system that we are implementing is that you can actually adjust these values, and you can, as you know, step 4 of the steps showed, like having a play around and showing, seeing for yourself what effect kind of changing the values of these different constants would have. So in this case, the formation of the complex is faster than the formation than this kcat. So that's why there's this kind of lag time where complex is formed, but the product takes longer time. So depending on the relative values of those rates, then these dynamics might look different. So that's something that you'll see in the examples that we're going to run. So switching gears a little bit in terms of the system this is applied to. So of course you can apply this to a cell biology context and many other contexts like epidemiology and population and ecology modeling. So as kind of a fun example, there's a kind of published paper that looks at a zombie apocalypse model. So this example that we're going to run through today is actually based on this paper. So you can go and find it at this particular link. It's linked to from the exercise that we'll do later, so you can access this. But essentially, yeah, we're going to build a model of zombie infection over time and look at how the population of zombies increases and how that depends on the particular parameter values and so on. So going from kind of a generic idea of how a zombie infection might work, we need to kind of write down the specifics of this system. And the exercise that we're going to do is just like the diagrams for the previous examples. We're going to go into breakout groups of around 5 people per group and draw a diagram of this model. So this kind of gives you the facts about the system. So this is the step 1 of list everything we know. So there are 3 classes of what you might call the equivalents of the molecular species, but 3 groups of susceptible, which is ordinary people, the zombies, and removed, which are basically the people who are not in the other 2 groups. So the facts about the system and how it works. So susceptibles are born at some rate pi and can become deceased through natural non-zombie related causes at some rate delta. Humans in the removed class can resurrect and become a zombie at some rate zeta. Susceptibles can become zombies through transmission via an encounter with a zombie at some rate beta. And zombies can be destroyed when attacked by a susceptible at some rate alpha. So the idea is that you're going to draw a model of this system. So basically with boxes for each of these classes, and then you draw arrows between them with these rates. So I think hopefully I can get some help from the KI team here to get everyone into the breakout groups. But before we do that, are there any questions? I see there's one hand up in the audience.
Yes. I wanted to ask about removed. They are the dead people after zombies will eat them, or who are the removed?
So yeah, the removed are they're dead, or they've been the zombies that have been attacked. But if you're familiar with zombies, they can actually be resurrected as well. So you can go from removed to being a zombie as well.
Okay.
Alex, I also grabbed a screenshot of this slide really quick and stuck it in
that's
the
very
chat
helpful.
box. Okay, great. All right, and I can also tell all of our participants that Alex will also have the ability to kind of run around the groups and just kind of check in on people and answer any questions. Is that fair, Alex?
Yep, I can do that.
Okay.
So in terms of drawing the diagram, I think there's a whiteboard capability in Zoom. If that doesn't work out, of course, feel free to use a piece of paper and hand that up, hold that up to the camera, just whatever works. Hopefully the whiteboard is straightforward.
It should be, and for everyone, if you haven't used that function yet, it is, if you want to stop screen sharing for just a quick second, Alex, when you get into those breakout groups, when you go to share your screen at the bottom, one of the options is whiteboard. So when you're in those breakout groups, you just share your screen under the basic functionality. You can share desktops or your whiteboard. So if that is helpful. All right, should we give it a try? and then we'll send Leonard and Alex around to see how everybody's doing.
Wait, where's the whiteboard tool?
Oh, yep. So if you go to share your screen,
Yeah.
the option is in there. So don't do it now, but the option is there.
Got it.
Awesome. Okay, so we will send everyone around. I'm going to go ahead and open rooms now.
Really gain a lot of insight by doing that, and intuition. I think it's an underappreciated part of building a model, is gaining an intuition for what the model is doing.
Oh, absolutely. I think that was a really great point when I said it just seems like it's such, I didn't mean to minimize its importance, but just my understanding is that it's nothing more than this, doing this. But I think you're right. I mean, what you said is it happened to us. I mean, we got to the final step and then it's behaving weird because we hadn't gone back and looked at how robust every different parameter was and that sort of thing.
Yeah, absolutely.
Welcome back, everyone. Welcome back. I'm going to turn it over to Alex so that we can figure out what happened.
Yeah, thank you. So, yeah, hopefully you managed to get something down in terms of drawing a basic diagram with some arrows on it. So I don't want to pick on anyone or put them on the spot, but if anyone's happy to volunteer and show us what you drew and talk us through it, then that would be great as a sort of starting point. But if no one wants to volunteer, that's fine too. I can just carry on with the exercise. So, okay. So I will, in that case. Sorry, did someone say something?
What you could do it step by step. I would volunteer 0 to S at a rate of pi.
Yeah, that sounds about right.
Sorry, I didn't understand. What did you say?
I said, well, we have to, S is an entity and it's created at a rate of pi, we were told. So I think we have to start by writing 0 arrow to S with a rate constant of pi.
Yeah,
Correct?
that's right.
Yes.
So yeah,
But
I think we could go through and draw this, but in the interest of time, since we only have about 33 minutes left, and I want to sort of show you the implementation of this, I'll go ahead and pull up the diagram that's actually from the paper, so you can see how they originally did it. So I will share screen, and there you go. So this is hopefully you can see in the middle there is the diagram from this paper. So you have a production, they don't use this empty set symbol I mentioned, but they're showing production of S at a rate of pi. And then this is the natural death where you move from S to the removed group at a rate of delta. You'll notice that they're multiplying these rate constants by the reactants. So depending on the formulation, you might see this done explicitly like this. You might just see it written as delta. I think it's probably more common just to see them written just as the rate constants, but because this is kind of a tutorial exercise from this manuscript, I think they've done that explicitly. So this infection from susceptible to zombie is done at this rate beta, and then from zombie to being, when they're attacked, or go from zombie to being removed at this rate alpha, and then zombies are resurrected from this removed group at this rate zeta. So hopefully you had something that looked something similar to this. So if you did, then congratulations, you managed to do step 2 of the modeling pipeline, and basically got you've written down something that you can take and then go and implement, and as step 3, and then kind of explore the parameter space and the dynamics of the system, which is what we're going to do now. So we're actually going to use, as Lena mentioned earlier, a framework called PySB. So this is, you can follow along if you like, and actually run this code in your web browser. So you don't have to. If you want to just follow, watch, then you're welcome to do that too. I will paste. I just need to open the chat and paste to everyone. So there's the link if you want to open this up and try it for yourself. As I say, if you just want to watch with a coffee or a tea or a cocktail or whatever you like, then you can do that too. So if you open that link, it's actually a GitHub repository. So if you scroll down, you'll see this link that says launch binder. So if you go ahead and click on that, it actually takes a minute to load up. So you'll see this page that says binder. It will take a minute or two to load everything. And then eventually when that's complete, you'll see a page that looks like this. So when you see this page, then we'll go ahead and open this top one. So that's actually open pretty quickly. These are now the same, as you can see. So if you go ahead and click on, okay, that's not useful. Well, hopefully, because this is the outdated one, I think. Yeah, so that they expire after a couple of hours, and I had that one open before, so you shouldn't have that problem. When you click on the top model, the top file here, just left click on that, and then you'll get a page that looks like this. So as I mentioned, this is based on this paper, so if you want to go and read that paper, there's the link to it there. You can just click on that. And this implementation is actually based on one from a published online by Christopher Campo, and then I've done a conversion of this for this. So this is called a Jupyter Notebook, and basically it's a way of writing a description like this, but also you have parts of computer code, Python code in this case, written in line. So there are probably there are other frameworks you could have used that are more graphical. So although this might look a bit daunting at first with some code in here, the idea is not for sure that you're going to go from zero to understanding all the details of Python. Hopefully you won't need to. I'll just talk about this from a high level and point out the main components of the model and show you how you can sort of implement this and start to play with changing the parameters and that kind of thing. So we've kind of gone through the background, what the different three groups are: susceptible, zombie, removed, and the different parameters. So in the original paper, they use Greek letters for it, but I find it easier to keep track of these parameters by giving them names. So you don't have to remember what beta is, for example. I just remember K, infect is sort of more of a natural language description, but this kind of shows you what they are in this original paper if you want to refer back to that. So the short version, if you want to run this, is you just click on this kernel menu, click restart and run all, and then you get this pop up, and just click this restart and run all cells. And then after a second, this actually runs on a remote server, so it's running, Google's paying for it, so you don't need to worry about that. And you eventually get this figure that comes up after a couple of seconds, and this shows you the output of the model. So this is time on the x-axis, days from the zombie outbreak, and population on the y-axis. So what this allows you to do is there are some sliders here that allow you to adjust these initial conditions. So this one is the initial human population, the susceptibles. So I can click on this axis, and each time I click on this, it actually reruns the entire model. As you can see, it's pretty quick and gives you the updated dynamics. And I can change as well this initial zombie population. So if we start off with some zombies, you can see that obviously the number of zombies increases quicker than if you start off with 0. So that's kind of the jumping to the end and the output here and showing how you can adjust these. But I'll now sort of show you how you can.
Hey, Alex,
yep.
we've got one question from Eugene in the audience.
Yeah, please.
Yes. I just have one general question. The language on what the script is written, what is it? Python?
It's written in Python using
Python.
the PySB framework.
Okay, that's clear. Thank you.
So there actually, I've included in this link, if we go back to this page, there's actually an implementation that's just using differential equations as well. So if you want to compare writing out the differential equations by hand versus using PySB, then you can click through to that. As I mentioned, using this reaction-based formalism is equivalent to the ODE one, but I find it more natural to think in terms of these reactions. So I'll just quickly talk through this, and you can see how this works. So this is all pretty much boilerplate at the top. This is importing the various Python libraries that we can use. So you don't need to know too much about that in detail. You can just paste that at the top. This model command that creates the empty model, and then we input the various parameter values. So the birth rate in this case at the moment is set to 0. So we just got a fixed proportion of individuals to start with, and there's no more susceptibles being born, which is why if we look at this, the number of susceptibles is dropping over time, and they're converting into zombies, and the overall population is fixed. So if we wanted to change that, then the nice thing about these notebooks is that I can just go through and I can put in a birth rate of say 10, and then if I press Shift Enter in there, whilst in that box, it actually reruns the code and then gives me a new result. So you can see this susceptible population is now going up over time, whereas it was flat before. So you can see it's kind of quick. So if you want to change the assumptions of the model, then you can just change these parameter values and rerun it. So there's this birth rate, which was 0 originally, so I'll put that back. This death rate. So in terms of units, which is a question that comes up, PySB, like a lot of modeling frameworks, doesn't assume any particular units. The important thing is just to be consistent. So in this, we're using a death rate, transmission rate, resurrection rate, and this destruction rate that are all in per day units. You could put these as per hour or per second. If you're writing a cellular model, you'd probably be using per second units instead. But yeah, so it's fine to use whatever, just as long as you're consistent throughout the model. So we have this death rate, this infection rate, resurrection rate, and destruction rate. So those are the 5 parameters corresponding to these 1, 2, 3, 4, 5. So once you've got those parameters in, we implement the 3 classes. Because PySB is designed for molecular modeling, they're called monomers, but you can think about these as sort of the class of 3 different classes in the model. Then we need some initial conditions. So these are the initial populations. So in this case, we're starting with 500 in the susceptible group, and nothing in the zombies group, and nothing in the removed group. And then we define the dynamics of the model. So the first rule is that we have birth. So there's this none keyword, which is this de novo synthesis. So we're just saying that susceptibles are created at this rate, k birth. And then we have the natural death rule. So this is susceptibles converted into a removed group at a rate k death. We have this infection or transmission rule. So this is, if you think in a chemical model, it's more easy to think about the kind of reactants because you know A plus B, both A and B are needed, right? So you need to think about what are the reactants in the product. So we have a susceptible plus a zombie goes to a zombie plus a zombie, right? So the zombie is conserved in this reaction, the initial zombie that did the infecting, and then the susceptible is converted into a zombie. So you can think about this as this is the sort of the reactants and the products of the rule, and then this symbol shows that it's a one-directional reaction. And again, this is going at the with the k infect parameter. Mentioned in the description that there's this destruction where susceptible can be a zombie, but instead of the zombie winning, the human wins. So the susceptible remains, but this zombie is moved into the removed group at this parameter k destroy. And then the final rule is this resurrection rule, where we have you can move from the removed group to the zombie group, and then that operates at this parameter k resurrect. So that all of that defines, that's all you need to define the model. So you have your set of parameters, the set of monomers or classes in the model, the initial conditions, and the set of reactions that occur. So are there any questions about the model? Okay, great. So I'll just quickly outline this code about how the model is running. So you need to define a time span. So in this particular, in Python, you use this library which creates this this thing called linspace. It's just linear spacing between two time points. So it's from 0 to 5 days, and we're taking 1,000 time points. So when you do ODE modeling, it's in a set of discrete steps. So those are what you need to specify up front. And then you create the simulator. So this is this SciPy ODE. So this uses a Python library called SciPy and takes this model and time span, and then you're just running the simulation. And then that's all you really need to run. So you can see that the actual simulation part is pretty straightforward once you've got the model defined. It actually takes more code to do this plotting and the interactivity. So unless you're going to be using Python, you probably don't need to know this in too much detail, but essentially this is just creating a plot of we've got 2 lines on the plot, one for the living and one for the zombies, and x-axis label, y-axis label, and a title. And that's pretty much it. All of this is just to add this interactivity to make this plot interactive and adjust these sliders so you can change the initial population. If you're doing this yourself and you don't need this kind of in the browser interactivity, as I showed before, you can go ahead and change these parameters, all these initial conditions. So if we wanted to start with, say, 100 in this remove group, then I can go into this and press Shift Enter, and then you can see that changes the dynamics as well. So that's it. That's basically the process of we went through. We had a list of the specification for the model, drew the diagram, specified the rate constants for this model, and then based on that, you can go and implement it as in PySB in this case, and then we ran a simulation for this model using these parameters and initial conditions, and plotted the results. And as I mentioned, the step 4 of the pipeline is you can kind of play around with this and ask questions about what if we had some zombies to start with? How does that change things? And you can see that the zombies take over much quicker. So yeah, it's just a nice way of, you can sort of ask questions of the model and say, what if we change this and what if we change that? And although some of these questions might be obvious, like, if you have some zombies to start, then they're going to take over quicker. When you get to bigger models with more complex dynamics and multiple players, then it's not always kind of as trivial. And that's really where the modeling can come in, and you can start to get, as Leonard mentioned in his talk, sometimes you can get to the surprising results that can lead to novel hypotheses and the discovery of new biology.
Or if there was a vaccine against zombies and only some people would take it, that would make it more complicated.
Yeah, if we wanted to make it more topical, then you could start to bring in these assumptions. And if you're kind of
I have a question though.
Yeah.
Serious question. You said you pointed to one line towards the very end. It said something about, and then we import this Python model to run or something like that. Mm-hmm. I didn't understand that. I thought you had defined the model with the initial conditions, the reactions. What was that bit about importing a Python model to run this? What were you talking about?
So this section defines the model itself, and then the next step is just to run the simulation in the PySB framework, define this time span. So it's not importing the model. If I said that, then I misspoke. But you create this this simulator object, as it's known in Python, and to do that, you give it the model, which is just called model in this case, and then this t-span, which is the time span, and then this creates the simulator object. And then all you need to do is just do sim.run, and then that runs the simulation. So this is the part that just does the actual simulation of the model.
You're just naming it, this name pertaining to this model and this time span.
Yes.
That model So the one thing, so this is part of PySB, that model command that Alex showed up above,
Yeah.
that automatically created an object called model with the small m.
Mhm.
Yeah, that's where that comes from.
All right.
Yeah.
Yep, that's right. So yeah, this automatically creates this lowercase model, which we're then supplying to the simulator. So it's just in case, you know, if you had the situation where you wanted to compare two models against each other, then you can give them different names and create two different simulators and compare them against each other and so on. So yeah, it just enables those kinds of use cases. But in general, you can just have one model and call it model. If you only got one model, if you're only looking at one model, then there's no ambiguity, and then it's all straightforward. So as David kind of alluded to, there's all these different extensions, and actually go into some of this in the paper as well. If you're interested in kind of delving into this a bit more, they talk about extending the model with latent infection. So there's a delay between when you move from susceptible into being a zombie. And I think as well, yeah, there's a model with quarantine as well. So, you know, you can see possibly this is, there's some topical aspects to this, and a model with, they actually have treatment rather than a vaccine in this case. But you can see how the kind of the model is extended. And, you know, if you wanted to, as an exercise, go through and extend this, then you can actually add these as extra reactions here. And that's one of the things that you'll probably find is that, I'll quickly show you this, this case here of the, this is basically writing the same model, but writing it directly as these equations. So personally, I find this a little bit more, the meaning is a bit more obscured here. And if you want to extend this, then you might find yourself having to update multiple different of these equations when you put in a new reaction. Whereas here you can just put in a new reaction in a new line, and the differential equations are generated behind the scenes and taken care of. So both are, you know, you can do both, and particularly for small models, you know, this is pretty manageable. If you prefer to deal directly with the math, then you can go ahead and do that. And if I actually run this, then you should see it's you know, it runs exactly the same. And this is not using PySB. This is just using the SciPy library. So, you know, you can compare that and show and see the difference there between the two. But yeah, as mentioned, you can sort of go through and extend this if you wanted to add and play with these different extensions about adding treatments and impulsive eradication, as they call it, where you have yeah, if you've seen a zombie movie, you know how this goes. Weapons and decapitation and so on. So yeah, you can extend the model with those kinds of things as well, and see how those change the dynamics as they show in these plots, and see what kind of levels of impulsive eradication you would need for the susceptibles to beat the zombies. So yeah, I mean, that's where it's useful, right? Although this is kind of a facetious example, you can imagine in a real disease scenario, you can think about, you know, what level of inoculation do you need, or vaccination, or treatment, or quarantine, and how these things would affect the dynamics of the different populations. Yeah, I see you have another question. Go ahead.
Yes, I was interested if you're going to comment a bit about putting the real measurement data into this model to improve it, like test its
You mean the calibration step of
yes, calibration step. Thank you.
Yeah, so I wasn't going to go into that today because it's a bit more involved. You've got to use some mathematical optimization libraries to do that. And unfortunately, I don't have any real life data on zombie attacks. If anybody has that data set, then do send it on. But yeah, I mean, for if you're interested in kind of calibration examples, there are, or the PySB framework in general, there's a bunch of PySB tutorials here that have some more cell biology examples. So I'll paste that link in as well. And again, it has one of these launch binder links in here. So if you click on that, do I have that open? Hopefully that opens fairly quickly. But so there's some tutorials there that go through some cell biology examples, and including one for extrinsic apoptosis, and I think there's some calibration data available for that as well. And if it's in that repository, I'll take a look when this opens. But yeah, if you're kind of interested in exploring the computational modeling more, then definitely that second link is a place to look, and you can launch this binder and run these different examples, and they've all got these various plots available, so you can see try this out on some different models.
Okay, thank you very much. I think I will do it later.
Okay. So there's one example like here. There's this catalysis model. So I mentioned this Michaelis-Menten. So I won't step through this in detail, but you can see this is one of the examples. If you click, so yeah, it's loaded now. So there's example models and tutorials. This is actually a tutorial that we've done in different contexts. So that if you go into tutorials and then catalysis tutorial, that's this one. And then, yeah, so this has the dynamics I showed in the plot earlier. And as David was asking about kind of whether that had specific parameter values, you can see those are listed there together with the initial concentrations of enzyme and unbound protein. So this example is a little bit more involved because it involves binding, and there are states. So this protein can go in the substrate or product forms. So yeah, it gives you a little bit more detail about how you would apply this to a biological context. But essentially it works in the same way. You're defining the same parts of the model, and then we're just running this simulation and plotting the results. So yeah, feel free to sort of take a look at that if you have more questions about how to apply this kind of framework to a biological example.
Do we have, can we ask questions?
Yeah,
I don't know
please.
if you can hear me. You can, okay.
Yeah.
So was this developed in the context of a course? Is it meant to be educational material? And is it all available?
So the zombie apocalypse example is based on this paper, as I mentioned. So you know that's linked to at the top of here is Muntz et al. So if people are kind of interested in learning, the goal of this paper is to teach how ODEs work. So I'd say this paper is a great place to look if you're interested in learning how to apply differential equations to a system. And I think it's a good example for what Leonard said about you kind of taking a system and then kind of formalizing it into a set of assumptions, drawing a diagram, and then this paper also goes into detail with the math. As a cell biologist,
were you asking about PySB?
Yeah, exactly.
About
I'm sorry.
the software itself?
The ability to run it online.
Yes, so the PySB software itself, I'm sorry if I misunderstood the question. So yeah, you can, at that second link there, you can actually run PySB software and all these examples online without having to install it, and it all runs in the web browser using this binder tool, which is a really great way to try out software. But if you're interested in trying it yourself, you can actually visit pysb.org. And
so PySB is an open free open source modeling framework. So yeah, it's like free to the community, and it's meant to do, you know, serious, you know, complex as well as simple modeling. Anybody can use it. And then there are, you know, what Alex is showing is a lot of tutorials. We have given presentations at different workshops on how to use PySB. So there's lots of examples and tutorials and interactive workbooks, that kind of thing, to help learn how to use it.
So this whole zombie story is very much based on the SIR model, right? And all the extensions of the SIR model. So I've been using something very similar. When COVID came about, I decided to teach a first year co-chem. So these are, on some campuses, they're called freshman seminars. They're 1 credit hour lightweight courses. And so, it was all new. So I quickly put together something like this, which was also based off of, which was also based off of another first year exercise I did in organismal biology that was based on the regulation of red blood cell, the production of red blood cell by the hormone EPO. But the students engaged so much more when it was about COVID. It was a lot more intuitive and to understand, how you get infected. And so some of the parameters they were varying were what if people are socially distancing? What if socially unmasking? What if there's a cure for it? What if there's a variant that's more transmissible and so on? So they kind of went through all that modeling and they were quite rewarded and happy to see that, they can just model it instantly. Yeah.
Yeah, I think that's one of the great things about building those kind of models is that you can get very quick feedback about and ask those what if questions and sort of see that quantitatively right in front of you.
Yeah.
So yeah, I think it's a great exercise to do.
Does anyone have any questions for Alex or Leonard? It seems like we are nearing the close.
I do have a question because I was thinking about model, because the zombie thing is so like it's in your face COVID almost. But I was thinking about actually how I would do it. Most of it seems pretty straightforward, except for the R naught. How would you model 3 versus 5 or 1? I don't know how I would put that into the model, the infectivity rate.
Yeah, it's just the for us, we didn't really calculate, we just have the transmission rate, the probability of transmission times the number of contact people have with one another. So we didn't directly use the R value itself, but the R value itself is also based on the rate of transmission, the probability of transmission.
You had a probability that any given contact would yield an infection from an infected person. And then you just multiplied by that, how many contacts they have. Okay.
Yeah.
All right.
So, but we didn't use the R. So they actually asked my students to calculate the R0 based on the probability of infection and number of contacts. Yeah.
I think it's an interesting point about modeling could be made here is that for the first year of the pandemic, the site 538.com was publishing all the different model outputs every day. And there were at least 10 or 12 different models that they were publishing from all over different institutions. And they were yielding quite different predictions. So it's not all that straightforward like anything else.
It has something to do with the
turns
beta.
out that some of the best models were actually the ones that were not so much based on calculations, but more so on kind of fitting the curve to previous outbreaks.
Mm-hmm.
Right.
So I think that maybe we can end by thanking these guys. They're now, they've done this sort of thing before, but now they're exclusive property of the FYIM trademark. We will be using them again. Thanks, guys. I thought that was really great.
Great, thank you. Hope everyone found it useful.
Yeah, thank you.
Thanks, everyone. Have a great night. We will see you tomorrow morning. Just a quick reminder before you go, lightning talks are at 9 a.m. Eastern. Otherwise, we will see you in the main plenary at 11 Eastern. Have a great evening.
Right. Feel free to email us if anyone has any questions.
Thank you so much for that offer, Leonard. That is very kind of you.
Absolutely. Thank you.
Yeah, this was a great workshop, actually,
Right.
especially the first part.
Thank you very
thank
much.
you.
It was really interesting.
Leonard, I have one comment for you guys if you want to stay for just a minute.
Sure, absolutely.
Did everyone else